My name is Kelvin, so I am a staff scientist at EPFL. So here's a brief background of just where I came from and how I came to be using the Creoptix WAVEsystem. So I'm from Canada, and then I moved to Geneva around 2015, where I was working on signaling within plants. There was an urgent need when I was there to find another way to say validate and also to determine certain interactions that we could not see using traditional methods such as ITC or even by more simple methods like pull downs. And now once again I'm at EPFL. So today I'll share with you two different stories. One that I was doing in Geneva on UVB signaling. A second one will be on receptor kinases, so membrane proteins with extracellular small peptide ligands. This was done by Satehiro Okuda, who is now an assistant professor at the University of Tokyo. And all of the work done that I'll talk about today was done in the lab of Michael Hawthorne in Geneva, the University of Geneva. So my journey with the system started around 2016 in Waedenswil. Me and an ex-colleague of mine, Ulrich Ullmann, who is now in Vienna, we went there to take a look at one of their earlier devices. So I dug up some photos back then, and I don't think the system has changed that much in how it looks like externally. But essentially, the system was small. We saw the chips as Fabio showed before, and then here's just the port that the chip goes in. So at least on the exterior side, it was looking quite sleek, and it was very easy to figure out what exactly to do with the machine. And we used, at least on my part, I used this machine to quantitate some interactions and kinetics. So as Fabio mentioned, we were looking at mainly affinity measurements, but because the Creoptix wave also offered the ability to measure kinetics, we were also interested in that. And some of this work I show has been is in this manuscript and also in this preprint here, which you can look up for further info. So just a brief introduction to the project so you understand where I'm coming from. So in plants, plants have many photoreceptors that have to that they use to sense light, because they are organisms that cannot move. So they use photoreceptors to sense different parts of the sun spectrum, which tells the plants exactly what to do, essentially, when to flower, when to grow, when to make seeds, for instance. And one of these photoreceptors is UVR8, and it is the only photoreceptor known within plants that can sense UVB, so wavelengths essentially between 280 and 315 at least in terms for this receptor. And why it's so crucial for plants, you can see here very clearly in these two pictures. So essentially plants that are lacking UVR8, so this UVR8 null mutant, essentially cannot survive well under constant exposure to UVB in comparison to wild-type plants. However, in the absence of UVB, these plants all seem to grow in normal conditions. So this photoreceptor is required for plants to acclimate to UVB radiation outside. So when I started the project, what we were interested in was exactly how this UVR8 protein can interact with its downstream signaling partners to mediate this signaling response. So UVR8 in its ground state is a dimer, and upon absorption of UVB photons, particularly at 280 nanometers, this dimer dissociates into monomers, which reveals a binding interface. This binding interface is presumed to then interact with a protein called COP1, E3 ubiquitin ligase, through this binding interface, but also through a peptide motif. I first characterized the binding of this peptide motif to COP1, and that starts with that goes back to the title of the talk, using GCI and both ITC to characterize interactions for one system. So we started with ITC for here because we were interested in the thermodynamics of the interaction, so the delta H in particular, to see if different peptides perhaps have different energetics in how they bind. So we can clearly see through ITC that these peptides from UVR8 and also other receptors or transcription factors can bind this COP1 domain with fairly low affinity, let's say. At the same time, we wanted to see if there's increased binding between COP1, this VP peptide motif, and also the second main binding interface that is revealed upon UVB irradiation. However, a big obstacle for us at this time was the fact that making this UVR8 protein was very difficult, and the amount that we can produce was very limiting. And so doing it via ITC was not really feasible on the long term. At the end of the day, for at least a few initial experiments, I could do these do the ITC experiments. However, this required vast amounts of materials just to get one simple binding curve, which we can see here compared to the peptide binding data. We can see here that the UVR8 peptide to this COP1 domain binds once again 190 micromolar. This is the same data as I showed in the previous slide. However, once this is the intact context of the full-length UVR8 protein, essentially there's a 1,000-fold increase in affinity to 140 nanometer, which suggests that there seems to be navidity or additional interactions between UVR8 and COP1 than just this peptide motif. However, to continue to probe this interaction, we required a different method, and that's how it led us to Creoptix with their WAVEsystem. And after, I would say, wasn't that long, so after a few days of just troubleshooting and finding the right conditions, we could easily, not easily, but we could get nice kinetic curves, which I show here, which display pretty much comparable, or I would say identical affinities to what we measure by ITC. So 140 nanomolar by ITC and 158 nanomolar by kinetically by the wave system. However, that was very happy for us to see, but on top of that, we could also characterize now the kinetic parameters. So we could say, or we could hypothesize to the speed of the interactions between the UVR8 and also COP1. But the main point I want to emphasize here at the end of the day, at least on the technical side, was that for the ITC experiments, I needed 200 microliters of 15 micromolar of UVR8 for just one run. However, for one set of data on the Creoptix Wave system, I only needed 300 microliters of 2 micromolar of UVR8. So a substantial reduction in the protein consumption, and that was also very advantageous, so we could do more experiments. A big advantage as well with the Creoptix Wave was in parallel with ITC, we could detect some interactions that we could not see by ITC. I won't go into further, but ITC requires heats that you need to detect to see an interaction. If the interaction does not give off any heats, either endothermically or exothermically, you cannot derive a binding curve, nor then a binding affinity. However, on the Creoptix Wave, this interactions are not dependent on enthalpy, and thus we could quickly characterize a mutated form of UVR8 which lacks this peptide motif, and we could characterize its affinity to say that it binds weaker than the wild type. So this was a great advantage, at least in our former lab, because we did not need to search for a different technique that was available in-house to confirm orthogonally something that we knew was happening, say, in vivo. And that's just the just a summary of the data that I presented. Yeah, as I said, you can see it on the two manuscripts here. How GCI really aided this study of mine was the fact that it was saved quite a bit of money in terms of production of the protein, because we were producing this protein in insect cells, and for every 5 liters or 5-6 liters of production, we could only get 1 to 3 milligrams of protein, which was not enough to do many ITC experiments if you needed to compare mutants and also have controls. But for GCI, we could do, on the Creoptix Wave, we could do multiple experiments because of the low requirements of protein. For both components, actually. As I mentioned, the Creoptix Wave by GCI, we could also complement other biophysical techniques that we had in the lab, like ITC. The main advantage here was we could detect binding for a mutant that we could not see by ITC. But for ones that we could, then it's just an orthogonal conformation, especially when the affinity constants were pretty much identical. And a big advantage also was that after seeing the machine, using the machine on site during the demo, and also once we got the machine, it was not that difficult to scale up our analysis to move on to other systems. The software was quite easy to use. As mentioned by Fabio, we did not really have to worry about our samples that much. If something happened, we could just dispose of the chip. It did not necessarily have to affect the machine. And also we had very great support from Creoptix, from the application scientists, in particular a previous Fabio that was with Creoptix. So that was very nice to do for my part. But I also want to share another story that was done by Satohiro. This is more, I would say, that really convinced us to get the machine in the first place back in Geneva, because the main focus of the lab back then was still with receptor kinases in plants. So receptor kinases in plants are very similar to other membrane proteins that are found in also other eukaryotic systems. So they have an extracellular domain that can sense extracellular ligands, such as hormones, peptides, small molecules from the environment, which would then trigger an internal kinase for downstream signaling. And this coordinates growth, defense, and many other pathways within plants so they can grow properly. You can imagine the same way in eukaryotic systems in mammals. In plants, in addition, signaling typically requires a co-receptor, shown here. So the kinetical system that a lot of researchers were studying, have been studying for a long time, was the BRY1, the brasinolide LRR receptor, which binds to its co-receptor SERC1. And its ligand, BRY1, is a very, very small hydrophobic molecule called brasinolide, which binds in this very small binding pocket. And when we first tested the Creoptix Wave, this was pretty much the data that Ulrich wanted to see, because we were looking to see if we can, finally quantitate the affinity of BRY1 and brasinolide, which was never shown before in, let's say, a biophysical assay, other than through, say, scintillation counting, through radioactive brasinolides, or through in vivo assays through just application of brasinolides to see the response in an in vivo context. But for us, we were more a biophysical lab, and we wanted to know the precise affinity and also kinetic rate constants and how this small molecule can attach to a receptor, which is very similar to what most pharma also want to determine between a drug and its receptor. And we were very pleased to see this, in particular because of the size of BRY1, which is around 100 kilodaltons unglycosylated, but it is glycosylated, versus the size of brasinolide, which is around 500 daltons, which is quite small. So it shows off really the sensitivity of the machine. After we could show this interaction, we wanted to move our analysis further using other receptors, and in this case now with peptide ligands. And that's what Satohiro did in these two publications that show off the work that was done on the Creoptix Wave, one in PNAS and one in Science recently. And I just wanted to show off something in the beginning. So like many other surface-based systems, SPR or BLI or GCI, you need to always optimize your system. And this is more technical aside, but for people who are working with large receptors, extracellular domains, we found that what really enhanced signal and also the quality of the data was to site-specifically biotinylate your receptor via an AVI tag and then couple that using onto a chip via strep-avidin. And we believe this is much better than direct amine coupling of an extracellular domain, because as you can imagine, if you have something very big, in particular like this receptor, you can couple the receptor onto the chip in many different orientations or couple it multiple times, which then reduces the activity of a receptor, decreasing signal when it does see its ligand, but also the quality of the final kinetic curves. So after we, or Satiro optimized the assay format, he could quickly screen through many different extracellular domains. And I show this here just to show off his work, but also to say that your data is much better when you come with very nicely purified, homogeneous proteins that have been well characterized. And by that, it's so nice that it crystallized very well. We also have crystallization and crystal data to confirm that it binds its ligand. But after that, we needed to characterize exactly how it binds in both kinetically and both thermal through its affinity constant. And what I show here is his data that shows Schengen3, which is this receptor, binding to a ligand called CYF2 and a homologous ligand called CYF1. And you can see here he gets very nice, strong, tight binding of around 1.5 to 2 nanomolar. And because he optimized the system for this class of receptors, he could quickly screen through other receptor extracellular domains of receptor kinases with their cognate peptide ligands, but also other ones here, which display a diverse range of kinetics, affinities, and this just emphasizes what Fabio was talking before. So with one system, we could characterize something between 1.5 micromolar to 1.5 nanomolar confidently, but all of these in particular have very diverse rate constants. And also, this goes back to what I showed during my data, was that we use this in particular to compare different techniques to make sure that what we see in one system can be confirmed elsewhere. And so here I show you something, this is the previous one before, the Schengen3 receptor with its CYF2 peptide ligand. In the absence of DMSO, that run was done in the absence of DMSO, and you can see affinity constant of about 1.5 nanomolar. This corresponds very well with a previous report done in another lab by ITC of around 1.8 nanomolar. At the same time, we were also interested to see if this affinity constant changes. In this case, Satihiro did this by ITC. He added 5% DMSO, and if you just compare all of these affinity constants for CYF2, you can see there's almost a 200-fold decrease in affinity in the presence of DMSO. And he was interested in this because most experiments for this peptide ligand in vivo are done in the presence of DMSO due to the hydrophobic nature of the peptide. But I think this slide just highlights that the fact that even if you have one technique in-house, it's not that bad to try something else to confirm it, especially if you have different conditions and everything. Also for many scientists I know that are listening here, you can trust, but always be aware that the conditions of your experiment can drastically change what you measure. So don't worry that your measured affinity constants are different than what is published. And to finish off this section, after he optimized his assay, after he could see and confirm his peptide ligand binding, he went forward to do a co-receptor here in orange screen. So before he started this project, there was no idea the cognate co-receptor for Schengen3, like BRI1 had Circ1. So he could then incubate Schengen3, similar to what was described before with ABA type injections for receptors to save on the amount of peptide ligand. He injected that into the buffer before, and then he screened a diverse set of potential co-receptors, which he could then identify as another Circ receptor, so the same Circ receptor as the BRI1 co-receptor. And you can confirm this via mutagenesis of the peptide and also mutagenesis of the full receptor. So at the end of the day, through the WAVE system, he could confirm the peptide ligand, but also find a co-receptor, which would be very difficult through other methods due to their lower sensitivity, but also high protein consumption. And if you want to know more, once again, you can look up these two papers. I'm pretty sure you can see them when Jill and me post this presentation online. And if you have any questions, you are also welcome to email me anytime, or email directly the authors who did this work, especially Setohiro. So thank you very much, and I'm open to questions. You can either type them or